doc.
Before we do that, is there anybody or any group who wants to present and not get on the schedule?
Does everybody know where the schedule is? So there's a Slack, I think Danny posted the presentation schedule. It's a Google Doc. And it's just numbers. There are nine different presentations. So we go according to that order, starting with you can't divide. Nice. So is there anyone that's not on the schedule currently?
Feel free to add yourself if you're not on there.
Add yourself wherever. Okay.
And we have, well, as of right now at least, we have 10 presentations. We have like 3 hours. Not saying we should take all 3 hours, but you know, let's aim for like 10 minutes. But if you go over a little bit of running, it's totally fine.
Is there like an Apple?
There's HDMI. That's this one.
Oh, that guy.
You get if you get the working note, that's good.
While they're setting up, I'll also say, we did ask you for survey with sort of feedback, but we'd love to hear from you other things as you go home and you want to write us an email and tell us how this was, and whether it was helpful to you or not, or what we would do differently, or what, please stay in touch. Tell us what you think about this.
Anyway, so the idea with the project was basically to essentially break multi-site resting state analysis. But good thing we didn't break it. The idea was to see if regression for site accounts for the variance that's coming from the site differences, and whether that's enough to account for site differences in multi-site data analysis using resting state. And so what we've done here is essentially look at ALF maps from the CPAC pipeline from a byte 1 data, looking at just males with motion less than 0.2 mean FD, and just looking at whether we can plot the mean ALF for the 3 networks, default mode, central executive, and salience network, and see whether we can classify or even identify patterns that are relevant to either site or scanner. And so here's just a bunch of stuff where we've loaded the maps, masked the data and stuff. And here's the original plot. So each of the 3 colors are networks, and the values associated with each subject are the mean ALF values for a given network mask. And the y-axis is essentially subjects ordered by site. So if you see there are like sort of 5 clusters here, and we believe that they're sort of representative of site differences or scanner differences, so to say. And then we basically just regressed. So here's the classification on this data by dividing train and test. No cross-fold validation, no nothing yet, but just we were learning how to do everything in Python. We're all MATLAB people, so this is just a brief thing that we did. It seems like some of the sites have really good classification accuracy on the test sample, but some don't, which is probably because the samples are not equally distributed across the sites. And that's something that we're going to look into later. But for now, we did that, and then we regressed the site information and also the mean FD value. I don't think you can see the whole thing, but here we go. Yeah. So we regressed the site information and the mean FD also from all of the 3 network mean values that we computed, and then we use the residuals to plot them, and that's the regressed values. So it is normalizing, which is good. We're basically saying that if you're looking at mean FD, then regressing for site should be enough. And then we also looked at the variance. So we did the same test, train test division and a classification on it. And again, there are some sites which have really good classification accuracy and some that don't. That's again dependent on the samples that we have in the site. So about 50% accuracy right now, which is not bad, but not what we were expecting. There's more plots somewhere. We also tried the k-nearest neighbor. So this was this SEM classifier that we tried, and we also tried the k-nearest neighbors. So pretty much similar results. So better actually, 64% accuracy. So yeah, we wanted to just learn Python, and we did this. And then there are more plots that Suyash is going to show you about. Yeah, just the plots for each out value. The stuff that you sent on the Slack.
Yeah, sure.
Influence might not be.
So that's basically plotting by site here. And so we basically plotted the mean values by site. So this is before the regression again. So we're just playing around with the visualization stuff and learning how to do things in Python.
It's learning the visualization stuff. That kind of stuff, yeah.
Yeah, so pretty much that's what we did. Any comments, suggestions? We're planning to basically make this like an official project and do this in a more formal way where we're identifying site differences and looking at many different features and seeing whether, so clearly mean does get normalized. Maybe there are other features in resting state data, maybe connectivity or other things that do not get normalized by regression for site. So we want to look at more data, and we want to look at more multi-site data and look at other differences. And so I'm sure other people have things to say. That's what we did, basically, yeah.
Did you look at the actual eventual group statistics to see if the regression changed it?
No, we did not. But we will, I guess. Yeah, and then Suyash did some of the cool stuff from the MRAQC data, so he wants to show you what he did.
Okay.
I guess for the Abide data set that we were working on, so the Abide people have a simple script to download subsets of their data, and as another simple hack, we hack that so you can download by group if you want just the controls or just the patients. So we'll put that up.
So all of this is on a GitHub repository called abide_multisite inside the Neurohackweek. So feel free to go look at the code.
Just wondering, how many sites were there?
17.
50% is really good though.
But then some of the sites had really small sample sizes, and the division between train and test set was not good at all. So this was because we were learning Python while doing this. It was like a whole thing where we were like, well, if the project doesn't end up with good results, we're okay with that as long as we know how to do things in Python.
But relative to chess problem.
Yeah,
Relative
compared
to
to
just
random,
guessing.
like,
This is just guessing based on the paper, like how many
And
samples
the k-nearest neighbor works better than this SVM, so that had 64% accuracy. So
That's pretty good.
it's pretty good.
So what I did was I just played with MRIQC paper which just came like 2 weeks back. My hypothesis was in MRIQC paper, Oscar has tried to predict from the MRIQC output, like we get 68 metrics for a single subject, T1W image, and from that matrix whether the image is good or bad, and there was a label data for the same subjects. So my hypothesis was if we add motion parameters from the task runs and from the rest runs, and that might help me classify better, but apparently that didn't work very well. That's why I'm not able to show the results. And I was able to classify, the accuracy was just 56% after adding the motion parameters from the task runs. So the underlying thing is not just dvars and FD mean would help. Maybe I need to look into other motion parameters so of course add some more features to make a classifier which is in the paper 75% accuracy. So beat that benchmark which I set for myself. So I'm nothing to show, sorry.
But we did stuff. We did do stuff. We're kind of making up.
Next up, team cognitive ontology. And while the ontologer is set up, we'll just remind you that some of you started various machines on AWS and may have not shut them off. And if you're still using them, that's fine, but I will go in and revoke all your rights on Monday and turn down off machines. If there are machines that you don't need, turn them off. Please, download, commit everything, and push it to GitHub before I turn them all off, so we don't lose on that.
Yes. We'll jump in.
Cool. All right, hello. So we had the rather simple goal of figuring out what cognitive neuroscience is studying and how it all works. And it sort of launches off from earlier work, in particular by the Poldrak group who've done things like this. And we started from this idea of the cognitive atlas. So what is it that we even study? What do all these words mean? And we sort of know when we read the literature that there's lots of confusion about different terms or used in different ways. And we have a lot of implicit knowledge, but it's not very well documented. Any given person is probably looking at different subsets and interpreting things in different ways. It's sort of hard to figure out what's going on at a global level. And so the Cognitive Atlas was this sort of attempt, as far as I understand it, to sort of deal with some of those issues. And it was more or less like a curated sort of Wikipedia-type model of figuring out some sort of ontology. What are all the terms that we care about? What do they all mean? And how do they relate to each other? And so it has these lists of concepts, tasks, disorders, sort of community-driven. In the end, they have a whole bunch of listings. It's not super well fleshed out. So we started having a bit of a look at the Cognitive Atlas and sort of a little bit of what's going on there. So it's sparsely connected in the sense that we have all these concepts and it's supposed to map out how they all relate to each other, but the vast majority of them have zero connections, and there's actually very few that are actually well connected to each other, and stuff like that. And does anybody, we had a whole group doing all sorts of stuff, so people who looked into the atlas.
There were only 53 people who'd ever done anything, who'd added definitions, and most of the definitions were added by the people who were starting it in the first place, so it maybe wasn't as well utilized as it could have been. Yeah, and I think we have some figures maybe?
Yeah.
You have something to say?
Let's see, one, two, one, yeah.
Solid.
This is a different
Oh no!
You might need a different, I don't have the other adapter.
Can you just
There are HDMI adapters here.
So yeah, there's HDMI.
Is this thing?
You can plug in HDMI if you have an HDMI port.
You want this one?
Yeah, I think so.
All the powers.
Yeah. There's also some weird stuff that we saw that I'll just talk about when we bring this up. For instance, there were some entries, so they says that there's 220 disorders listed, but it turns out that you can't find those on the website. In fact, there's it's only 5 of them, but 5 of them were just fake entries that whoever built the website had left, flapjacks and wingnuts. And one of them had the definition. Did I do this? So it was a little funny actually getting into the API. I feel like it's probably hasn't been used as a resource as much as it could have been.
So
going back to the main part.
Yeah, so sort of the goal then was to go about this a slightly different way, more of a data-driven approach. And so can we sort of derive or analyze this kind of approach through the literature? And it sort of inspired similar to things a little bit like neurosynth and some of this stuff, but not necessarily restricted for fMRI papers. And so we're actually just jumping on top of the PubMed API. And we're going to start with the sort of project outline that we were working with. Is we're going to start with the Cognitive Atlas. We're going to try to sort of figure it out. We're going to take their terms as a base set. try to mine them, their connections and their definitions in some sense, from the literature itself. And then we can do two things. One, we can sort of check whether the literature supports what is sort of outlined in the Cognitive Atlas, insofar as the Cognitive Atlas is outlined, and sort of see differences and similarities. If they're roughly similar, the data-driven approach would be able to sort of complement or sort of propose new connections and so on. And insofar as they're different, it would be interesting to see where sort of this manually curated thing is missing things that seem to pop out of the literature-driven approach. So to do that... Sorry?
Can you zoom in?
Yes, sure. So to do that, I had actually done a little bit something similar to before. So we worked on sort of generalizing some code for a very specific type of PubMed searching to more of a general library for searching PubMed, given these search terms. And then we sort of do a couple things. One is which just seeing how they map together. So checking the literature for co-occurrence. So given terms of interest, how often do they occur together in the literature? And these searches that we're doing right now are all based on titles and abstracts. So it's sort of a coarse sampling over but a very, very broad corpus. And that kept timing out, and I don't have the counts data, but we did scrape a bunch of the words data. So what I mean by that is, given these search terms, going through and finding all the papers. The search terms are just, for us, they're all the concepts. Oh, I'm not this computer. They're all the concepts from the Cognitive Atlas. So take those, find all the papers that mention those terms, grab all the abstract data, title data, and metadata, and we can sort of characterize and profile these terms based on the literature that is made about them. You want to say a little bit about some of the words analysis we did?
Yeah, so basically what I did was, starting from the fact that some of the data had already been scraped from PubMed, and we had a list of abstracts that are associated with a given keyword. So the first step was just to kind of create a corpus out of those lists of words. They were in the back of words format, so really suitable for NLP analysis. And we implemented some really simple things, but very telling, about something like cognitive concepts in literature. So I can just show you the output here. I'm not going to run it from scratch because some of these steps take a little bit longer. But we are using an example case of when the keyword that people are looking for is autobiographical recall. So we have a JSON file that contains all the words that appear in the abstract associated with autobiographical recall. And there were 86 articles in this case for this phenomena, because it's kind of like a interesting term. You wouldn't find it in literature very commonly. And then we can get, like, the user can provide the number of terms that they want to look at, which most frequently appear in the corpus itself. And these are here. And I think this is an interesting example because autobiographical recall should be the most common word when you're looking for that phrase, but actually memory is even more common than the word recall when the phrase you're looking for is autobiographical recall. So that's why I think it was sometimes useful to retain information about the keyword that you're looking for. And then you can also use co-occurrences within the text to find out what other words are similar to the keyword. And in this case, they were all more or less close to memory and semantic memory. And then we also played a little bit with topic modeling. So just from the abstract test, just restricting to a small number of topics and visualizing what the factor loadings were within those topics. It's kind of hard to get at a good visualization of this kind of data. We were thinking about doing a little bit of hierarchical clustering of the terms and stuff like that, but we didn't get quite there yet, but it's definitely in the future goals. But the point is that we get a lot of data from scraping all these abstracts, and it's really, really easy, given how you've implemented our code around the PubMed API, to just jump into NLP straight from there. And right now it's just in the Jupyter notebook, but we'll definitely write a module out of this or something so people can use it if they're intrigued.
Yeah, cool. So I guess, do you have the GitHub?
Yeah.
Just show you. Most of the stuff is sort of on, in terms of like, so there's Neurohackweek data-driven cognitive ontology, sort of lays out a fair amount of the sort of what we're doing here. So we didn't really get as far as actually comparing sort of a data-driven versus this curated ontology. That sort of stuff we'll sort of continue with. And there's all sorts of like tangential questions we go on from here. But some of it is fairly well set up. So there's a little library that you can run these analysis, and it's pretty agnostic about what you're searching for. So you can just put in any sort of terms list if you want to play around with like counts data or scraping this sort of data. And then I think this is live. Nope, not this. So here we can sort of just plot some of we can pick terms of that we actually scraped. So for example, attention, and grab all this data. And this is part, similar to what we were doing here. And you can just scrape, what are the words most common for attention or your favorite search term. So like we could go back to, what was autobiographical recall, and then see this is another way to sort of plot these things out. So there's fairly sort of tractable way to explore some of this data as well. And yeah, so we were all sort of poking around at stuff that's not really our main area in terms of text mining, APIs and stuff. So we had a bunch of fun. I don't know if you guys have. And we all did lots of different pieces that came together, exploring the cognitive atlas, exploring the text mining, exploring, just figuring out what this project means and so on. So it's probably something we'll try to keep poking at.
Cool.
I think that's sort of it.
Questions?
Yeah.
When you scrape the words, is it only from the abstract, or do you actually scrape the content of the paper?
Right now we're using the PubMed database, which is searching for titles in abstract and scraping titles in abstract. It's a fairly straightforward change to use PubMed Central, which has full text, in which case you can search full text and scrape full text. It's not what we're doing right now. But it is a subset of papers available through that. As far as I can tell, you don't have to do anything with PDF parsing, so it's pretty friendly. And although it has many, many fewer papers available, you can search more broadly. So right here we're doing concepts which are likely to be mentioned in abstracts. We also have like a task list. We were trying to, so Cognitive Atlas, for example, has this mapping of which tasks record, sort of measure which concepts or which categories of things. So we also wanted to sort of reconstruct that from the literature, but you couldn't necessarily do that from abstracts. So we're pretty sure it'll generalize pretty cleanly to a full text analysis, in which case you could get methods, sections, and so on. Cool.
Yeah, he's Arthur. What are the rest of us? If you want to start plugging in.
Okay, so while Dan prepares everything, so we were trying here to build a module in Python to be able to have these avalanches in a certain way that would be easy to visualize. And well, as I was telling on Tuesday, these avalanches are just a natural phenomena that could be modeled to be seen in the brain. So we were like the first day discussing how this model has many constraints, and you have to be thinking about many ways of actually building the avalanches. So we finalized having some basic steps that are the following. Like the first time you have to look for extreme events, like having this threshold over the whole data set, and just binalizing extreme events over a given threshold, and everything under zero goes to under the threshold goes to zero. And after this, there is a step of clustering in both spatially and temporally. So the avalanches have to spread and burst over time. And finally, we were discarding single voxel clusters and clusters that don't last more than one time point. So this was like a cleaning step in the code, and that's all. Yeah, we have some code and I'm close to solve. That's it.
Okay, that's just to illustrate what Azir just told us. I mean, we just had this Python notebook where we did exactly this. So we had some preprocessed data, which was filtered and then registered to M&I space. We tried to extract this point processing as we showed there. So we tried to point process the entire array so that we have only zeros and ones of the entire time point. And using this, we detected some clusters. So basically the major challenge there was so that we tried to, how could we make this efficient, how could we cluster the entire time points in an efficient way, and how could we get a visual and a nice way of representing the entire time points, what we're doing in an efficient and informative way. So that's what Daniel's going to show us, so nice pretty plots and colorful plots of the avalanches.
So, come plug in. So this is how's everyone say going? Okay, this is how this is the paper that this method was first introduced with. This is how they showed their avalanches, their 40 avalanches, and I think, as was pointed out on Tuesday, that leaves something to be desired in terms of being able to see what's going on. So if I make this smaller, this is the wrong one. Small. Oh no, I can't move it. Okay. I can't get to the maybe I can't get to the top of the window right now. Okay. Now I can do it. Okay. I'm gonna bring it down. It's pretty cool, I think. So the idea is that these are clusters of above threshold activations that are contiguous in space and also continue in time. So kind of like an avalanche, the idea is that you get this high magnitude event that kind of spreads throughout the cortex and subcortex. Okay. I'm just gonna go to that one because that one's smaller. I can play. Okay, here. Well, it's playing at least. So you can see they kind of come online, and we've colorized them. And if I can just kind of scroll down a little bit. Yeah. They seem to be kind of following what we think of as kind of anatomical and functional boundaries. Kind of bilateral hemispheres will kind of come on at the same time. In like sensory areas, and this is all post hoc and harking, and take it with a grain of salt. But it seems interesting, at least visually. And what Brian worked a lot on is actually trying to reproduce some of the science that was in this paper, which looks at the actual distribution of the sizes, essentially how many voxels are in this continuous activation. And long story short, from it, given certain theories of how the brain should work and criticality and physics and all that is a lot over my head. You should get this kind of slope, this power law slope of sizes, in sizes of these avalanches. And that's what this paper claimed, and we can actually show the same thing in our data, which is cool. So this is a log-log scale of a probability of an avalanche at log size. And that's kind of cool. And so we've got it up on GitHub with code and anything if anyone wants to play with it. Yeah.
Do we have any other example with bigger avalanches?
Yeah. So the one that I can't get to work is come and find me. It's cool. Like eventually, like every now and then, the whole brain kind of lights up. Which, if you ever kind of looked at an EPI time series, there's a couple of times, every time series where the brain kind of all gets
bright and then all gets dark, and that seems to be important, like up-down state sort of thing. Whether this actually is doing anything scientifically useful remains to be seen, but it was fun.
Team Angelina.
Yours. I'll start and then you go?
Yeah.
Okay. Okay, cool. Hello, we are Team Medellina. So we're all neuroimagers and we all want to study the brain by making measurements of it. But what happens when we start to collect more and more data is that our measurements get less accurate. So our idea was to create a crowdsourcing game to get human input from medical image segmentation and combine that with machine learning, eventually, to get the most accurate measurements of the brain. And so with that, Dylan can start.
Yeah, so we've got a little bit that we'd like to tell you guys about, but first, I want everybody, most of you have your computers out. If you don't have your computer out, take out your phone, take out a device. I know you'll have them. I've seen you on them all week. Go to medulina.com. Sorry, I got a million slides open. So it's just this, medulina.com. And if you can't get there, raise your hand so I know about it, but we're not going to try to fix it. So go there. There's and let's see what that looks like. So you should see something like this. And so that we've got an easy, a medium, and a hard. While we're doing the rest of our stuff, I want you guys to basically ignore what we're saying and play this game. So pick easy, medium, or hard based on how good you think you are at tracing things on brains.
How all the planes work?
Yeah, sure.
Okay.
So pick so you're going to log in with GitHub, and then it'll show you a picture of a brain. You can, if you're on your phone, you can use your finger to draw, 2 fingers to pan. If you're on your pinch to zoom. If you're on your computer, you can just click. And so you're going to outline the tumor, and then double tap or double click inside it to fill it in. Oh, I missed a little bit there. If I, whoops, you can hit this undo button. If you want to see what the brain looks like underneath your trace, you can press that. You can usually press that. And then you can submit it, and it'll tell you how you did. The red is what you missed. I didn't miss anything. The blue is what you traced but shouldn't have. And the purple is what you got correct.
Which one is contrast?
Oh.
Yeah.
If you want, there are also brightness and contrast tools that Anisha would like you to make sure you know about.
Yes.
And the score.
Oh, right. So you get
A dice coefficient.
it'll tell you about that up at the top, and you can press next to move on to your next image. It saves your brightness and contrast settings. And then a little bit later, we'll tell you about some other features. But right now, trace brains. All right.
How do you feel the...
Double click or double tap if you're on a phone to fill something that you.
Do you want an iPad, JP?
I like my computer.
Okay. And so now while you guys are doing that, I'm going to do a short play for you. This is a brief tale of live user feedback. So the other day I asked my wife, Kaylee,
to let me present this. Asked her if she had tried Medulina.com yet. And she's like, oh shoot, I forgot, I'll do it now. I was at the grocery store when you sent it to me and I forgot. This is hard, I'm not catching on. Everything the website says is correct, looks like nothing to me. It's 5:57 p.m. Some of them are pretty hard. We'll give you 2 minutes and let me know what you think. Yeah, I'm gonna complete it. I'm just getting them all wrong. So there's not a thing you complete. We've got a ton more images up there, and we've only actually, since we're breaking the brain into slices, we've got a ton of images out of every brain. We've got like 3 brains up there for some of these tasks, and it's hundreds of slices. So she said she'll do it for 10 more minutes. Then we're having some technical issues. The internet is super slow. This was great user feedback. We've since fixed some of the image size issues that were causing things to be so slow. Trying to get it tried on different devices. And then she's like, oh, I'm getting better at it. This is 9 minutes later at 6:02. So that's great. 6:12, so she tried it for 20 minutes just because she's such an awesome wife. And she said that it was hard. And then I told her about the leaderboard. And I'll show you guys the leaderboard in just a second. And then 15 minutes later, she's still playing. So this is 6:30, so she's been playing for 45 minutes. And she's like, oh, this is fun. I'm going to play all weekend. And then she's complaining about our leaderboard being a little bit bugged, so we went and fixed the leaderboard bug. And now she's catching up to me because half the time when I'm doing it, I'm debugging something, so I just draw something real quick. So my score has not been great recently. And then I asked for her permission to share her feedback with you guys, and she gave me her permission. So I'm not going behind her back or anything. But yeah, so that was some live user feedback. And then if you guys would like to see the leaderboard, we'll show you later. Keep tracing. So some of the things that some of the other things that we implemented over this week are very nearly finished researcher dashboard. So Kaishi, would you like to tell us about that? Sure.
It works.
Oh wow, so this is live demo on live demo.
Yeah, no, for sure.
Oh, that's the wrong dongle. Does anybody have the old Mac dongle?
That's okay.
Or is that one of the ones that's up here?
Just unplug it. Just unplug the. Yeah.
Is there HDMI? Is there an HDMI? No.
Yeah, we have HDMI.
Perfect. Thank you.
Okay, so if you're a researcher and you want to start a new type of segmentation task, these buttons don't work, but these ones do. So this is the task type, slice direction, patient ID, the file name, just things you would need if you were trying to organize uploading a task for millions to do. And you can check and see what you've uploaded, what you haven't. You can search for something like stroke or a person who would normally be like patient 7, but and then if you have another file you want to upload, then you choose your there are 2 nifties. You choose your background image nifty and the mask that you have painstakingly had an expert outline. You enter a subject ID and you enter a task, like stroke. You say what is the minimum number of voxels you want to visualize so nobody's searching around for one single voxel in the slice, and then you submit it. And what it's doing right now is taking the nifty and it converts it into the little tiny slices that you see, and then this is just a QC page that pops up and asks you to check your training data below. Make sure you didn't upload anything incorrectly.
And so the idea is some people's institutions might not necessarily be comfortable with them uploading this information, uploading entire nifties. And so this is actually going to be containerized so that people can run it all locally, so that you can process all your nifties for upload on your own machine, and then you're just uploading a set of JPEGs that are completely stripped of all metadata and should be much closer to de-identified human data to make IRBs happy with releasing this sort of information. Oh, sorry. And then our last up and coming feature. That's one way to put it. last but not least, is that better?
Well, I mean, up and coming. That's a nice one.
Optimism, right? Yeah, so you bounce through those.
So, as we all know, deep learning is this magical thing that just works, right? And it can predict everything. And so I just want to bring this back a little bit to why is this data being collected? Like, why is it important? And it's obviously really expensive to get expert annotation on tumors. And so wouldn't it be great if deep learning just told us where the tumor was? And it should be super easy, right? I mean, we have like almost close to perfect performance on these like data sets. Like, you can just give the network some samples and it'll say it's a ship, not a dog. So it should be really easy. But the catch is, and you know, there's really nice architectures out there that use convolutional layers to find interesting features and not get overwhelmed with the number of parameters and feed it to a fully connected network to make a good prediction. But these data sets, aside from being like meta overfit, also have like 60 to 70,000 samples. And obviously MRI data is like really expensive. So the plan is to, and obviously it's not done yet, that's why it's just a plan. There are some cool techniques for using like pre-trained networks and essentially taking some of the features that they abstract and just retraining some parts of the network to your problem. So the idea would be to train one of these networks on larger data sets of like brain or no brain, or something that would allow it to pick up on like brain-like features, and then on a much smaller data set like the meningioma tumor data set, train it to actually detect the tumors, and eventually to add like an additional channel of information that would take the data that was collected from you guys right now, for example, and give it like stronger priors as to where the lesions or the tumors or the hippocampus is. So that's the plan. Leaderboard time.
So if everyone clicks on their profile picture on the top left, and then hit leaderboard. Okay, log in. Okay, you can see your score and you can see a nice animated D3 plot. If you click on one of the dots on the plot, you can see what you did wrong. So it kind of takes you back in time to the time that you did that. If you want to see the average for this slice of all users, you hit slice stats. And you can toggle between the truth.
Oh man.
Yeah, so you can see that for this particular slice, if we were to like threshold it in some way, it would be very close to the truth. So
the truth? Never mind, it's a bug, I'll show you later.
okay, yeah, there are probably some bugs. So come find me if you have any bugs that you can reproduce. And yeah, so hopefully as we keep developing this, get an IRB, and put this on Amazon Turk, we'll be able to watch and see how our users are doing on this task, and we can adapt accordingly.
So one suggestion,
yeah.
Sometimes there are like same slices, contiguous slices that come like complicated. You might want to shuffle that up.
Yes, so we do have
It's selected randomly, but the thing about random is that humans think it's not random.
Yeah, but
just as a suggestion, because we tried something like this before, the idea was to also get some online games to put a link on. So there are some games where you can either pay for assets, or you can actually go and do tasks like this and earn assets like this. You get a different population than on Amazon Turk this way.
Oh yeah, so definitely gamification is.
So what she's suggesting is for something like Candy Crush, where they say, watch this video to get more candies to crush or something. You could pop up a link that says, go trace some brains to get more candies to crush.
Wait, that's really cool.
And you get a lot of different users.
I like that. Yeah, click this, get candies for crushing.
Yeah.
Pokemon.
And then we can, so we need to set up a nonprofit so that Candy Crush could treat this as a donation in kind, and we don't have to pay anything for it.
That's complicated, but yeah. What are you saying, Zadra? Oh.
So when you will train the machine learning model to maybe give you a prediction for implementation, how are you thinking of incorporating the user feedback?
So we're thinking of having it as an extra channel. So you can put like RGB images into deep learning and it can, find things, but now instead of RGB it would be like grayscale image, and then on top of that like a human non-expert prior as a second channel. So hopefully you don't change too much in that.
Yeah, that's the easiest
network.
thing to do.
And then the next step would be no expertly traced thing, just using the crowd?
Yeah.
Right, so right now we've only loaded up the training images, but the idea is that once people reach a certain score threshold, they would switch over from being shown predominantly training images to being shown test images with occasional training images just to make sure that their accuracy is staying up.
Make sure you're human, all that kind of stuff.
Yeah.
And so for the user, would I like just upload, it would be like free, or would I pay for this?
As a researcher, I don't know
I'm
yet.
joking, yes, in advance.
As a player, you don't.
But as a researcher?
Oh, as a researcher, well.
I think as a researcher,
both sides.
Yeah, it's going to depend on how much the infrastructure costs us. But the idea would be that we can help researchers set up a task here and then make it painless for them to push it over to as a Mechanical Turk task.
So that's a company basically that we started. Yeah.
I mean, we did register.com.
Oh.
Do you plan to have some sort of 3D visualization so that you can, if you can segment one slice, most probably it's going to be very easy to segment the neighboring slices, or they can help you to segment it more correctly?
Yeah, that would definitely be the goal, but I think then you run into some of the issues that maybe you'll hear about later that like it's just a lot more difficult to fit in memory, but I think that's definitely
But
the
maybe
goal.
just like a neighboring, kind of like a slice through the neighborhood.
Yeah, and they do have 3D convolutional layers in these networks, so it's definitely possible. It's just like a little further ahead than we're at.
Or did you mean when we're having the humans trace it?
When the humans are looking into it, I mean, I guess that could help.
It could potentially help, but especially for Mechanical Turk tasks, there's a real premium on making each individual unit of work that they're doing very self-contained and very quick.
So show that at the same time, like show some other prior information.
Right. Yeah. But yeah, it's a good idea.
Yeah. You could also then have a VR game.
Yeah.
Whoa.
Yeah, if the next hack, if you'd like to code that for us, that would be amazing.
All right, cool.
Thanks.
Cool. Speaking of which, deep learning and preprocessing for brain lesion segmentation.
Yeah. You changed the order. We were.
Oh, sorry.
6.
Yeah, it used to be. Yeah, there used to be some duplications there. Sorry.
Okay.
So only.
Yeah,
I see.
Yeah, we should.
Yeah.
That has all kinds of.
I'm sorry, just one more question while we're setting up. Did it crash for anybody?
It did once.
Okay.
That was just you.
Alright. Yeah. It's like it's running on like a micro instance, so I was almost certain it was gonna burst into flames.
Is that arson if you knew it was going to happen?
Yeah.
Somewhere in Seattle, it's just the admins rapidly.
So, about deep learning. So the idea was to explore this kind of deep learning magic, and the motivation was exactly the same. So we know that sizes for typical samples of 2D images can be up to millions of examples, and images itself are pretty small. In opposite, for neuroimaging, we have large dimensionality and very low sample size. And I believe that we can still obtain reasonable results due to some kind of unification of our images. And the idea of the project was to explore effect of pre-processing, so some kind of unification on quality of deep learning segmentation. And so just an example, another important thing I had in mind that if we have data from different sites, from different tomographs, we will observe significantly different patterns in each. So here we have flares from 3 different sites and a data set for white matter hyperintensity. So you see it, here we have very bright skull, and here not as bright. And another idea was to use as many tools as we learned this week. So it's a huge technological stack. For cloud computing, the initial idea was to use CFN cluster and plus Docker. So CFN cluster, it's a library which allows you to launch SGE cluster in AWS. And the problem is that you cannot use Docker with SGE. So I thought that there was a security issue, and if I launch my own cluster, so I'll be fine. But the problem is that we cannot use Docker at all. So we have to convert our Docker images to Singularity. So, but we didn't do this during the week. So we have to split our computations into 2 parts. For neuroimaging preprocessing, we used AWS plus Docker, and we prepared a small wrapper for this. And for deep learning, we used Kubernetes plus again Docker. For neuroimaging, of course, we used NiPipe, thanks to great tutorial and thanks to Dave. And yeah, for programming, again, the whole stack, except maybe R. Yeah, so about AWS setup, probably it can be useful for you. So we have a small script which allows to set up your AWS instance for neuroimaging processing. So it's install all necessary software. It's Python, Docker, some basic Python libraries. And also it provides you a very simple way to launch any command, any bash command inside your Docker image. So like this one. In image, it's in the node itself, there is no Docker, of course. So no free surfer and no free surfer variables. Well, and I hope that we will also push these things with SGE in a couple of weeks. So, pre-processing.
Sure, yeah, so I fiddled around a little bit with NiPipe before I got here, but it was a great reminder. This was a terrific opportunity to try out some of the workflow opportunities that NiPipe provides. And so we put together a pre-processing pipeline that leverages, BET for skull stripping, ANTS for mutual information-based co-registration of T1 and FLAIR, bias correction from NuCorrect from FreeSurfer, let's see, M4 bias correction from ANTS, tissue segmentation from ANTS, et cetera. I mean, we were able to tie together all these tools at using exactly the kind of reproducible pipeline that NiPipe provides and supports, and all of this feeds back into a data sync, which can then be fed forward into the deep learning algorithms that I will describe next.
Yeah. I'm not sure that it will work. Yeah, it works. So it's our instance on AWS clusters. So you see 40 CPUs. We tried to use 64, but it was all gone, so we cannot launch such a large instance. So it takes to process 60 images, I don't know, 10 minutes.
It was,
yeah, it was very fast.
So I was ready for it to run all night, and it was done in about 20 minutes.
Yeah. So about deep learning, the original idea was to fix deep learning architecture and learning methods, training methods. So we used these segmentation. This architecture for segmentation was developed a couple of years ago, especially for brain lesion segmentation. In that case, it was glioma segmentation. And so the idea was to answer some various questions, like if we perform skull stripping, does it lead to improved quality of segmentation, or should we use isotropic resolution, because these convolutional networks, kind of isotropic network. So we had a lot of interesting research questions. But the problem is that deep learning experiments are still in progress, so we cannot share results with you. So it's Kubernetes, and my job's in Kubernetes. So it's recent jobs, but yeah, we don't have enough results to say you how to process your images if you want to segment some kind of lesions. But I hope that we will, in a couple of days, we will receive all results. And so if you are interested in, just let us know, and we will share the results with you. So another side issue of this week is that we made public a library for segmentation of mainly neuroimaging data segmentation, which is under development in my group. It's, in my opinion, it's very well designed and very badly documented now, so it's impossible to use it because there are no comments, no signatures, the definition. Some readmes are in Russian, but it's really highly modular. So it's right now it allows you to simply change the data set and launch several models. It's just one of them, and now guys feel some kind of pressure because it's not kind of private library, it's open to the world. So they already opened an issue for refactoring and some kind of stuff. So I hope that in a couple of months it will be usable for segmentation, for general segmentation. Yeah, that's all. Thank you.
As far as I know, they don't make the trained models available. They make the model architecture available.
Yeah, we have implementation of several popular segmentation methods. So we implemented DeepMedic using TensorFlow, as well as UNet. It's for more biological image segmentation, and several popular general architectures like eNet.
I think the question was about the weights from your training. Right?
We share fitted models.
Yeah. I'm not fully understand the question.
So you were asking about the weights. Once you've trained it, you can store the weights and you can share those weights for others.
So it's public. It's a public library. So you can train your model. It's all models are written in TensorFlow, and then you can export the weights and use, for example, as some kind of pre-training for another task.
He was asking whether you and your group would make the weights that you found in your experiments. Maybe.
It depends. It depends on experiments, yeah. But we can do this. We can actually. Right now, we don't have any publication on segmentation, but it's a rule of thumb that we will make Docker code fully available, so it will be easy to obtain any kind of model or weights from these models. Yeah.
Any other questions? All right, predicting individual differences.
Okay. All right. So our team was interested in learning about different machine learning techniques and applying that to predicting individual differences. And we had a lot of fun. And Jeanette was on our team, but she's not here today. So she actually told us this. So it's a proved message.
All right. Yeah. So we had the kind of modest goal of seeing if we could, using the HCP dataset, task fMRI dataset, seeing if we could use machine learning models to build a neural signature of working memory, and then to use that model to predict in-scanner and out-of-scanner behavior, to kind of predict individual, or correlate, I should say, individual differences in behavior, essentially. So we're using the HCP data, we're using a working memory task just because it seemed like kind of as the first proof of concept, the best kind of task to use, robust activation, obviously for working memory tasks. So everything I'll present here is done with working memory, but in principle it can be applied to the other tasks in the HCP data set and obviously other tasks in general. So we did a couple different things, but this was kind of the most interesting and probably the easiest to present. So it's kind of a three-step, and that. There we go. All right. So it's kind of a three-step analysis pipeline. So the first step is what I'm referring to as building the neural signature of working memory. So the idea is we just, using a training set of 700 subjects, trained SVM to discriminate between 2-back and 0-back beta maps. So we did just five-fold cross-validation to train up an SVM that could predict between 2-back and 0-back. And that's what I refer to as the signature of working memory, is that trained model. And then we took that model and applied it to a test set of 300 more subjects, where we basically just took the weights in that SVM and took the dot product between the weights and the 2-back greater than 0-back beta map for those 300 subjects. And this is an oversimplification, but the basic idea is that dot product is essentially a distance to the hyperplane, to the fitted decision boundary. And then we took that dot product and correlated it with various forms of behavior. So basically the most kind of basic correlation we did is correlating with in-scanner behavior, so in-scanner working memory accuracy. We correlated it with an outside of the scanner working memory accuracy, like a list sorting task. We did a control analysis where we correlated with something where you wouldn't expect working memory neural signature to correlate, which is a personality metric. And we also did another control analysis where we trained in step 1, rather than training 2-back versus 0-back. This task used both faces and houses and places and other stuff. So we just trained on the beta images for faces and places, and then did all the same rest of the pipeline, so correlating with behavior and taking the dot product and all that. The idea being that there shouldn't be any correlation in your representation for faces versus places with any of the working memory metrics. Does that make sense? Cool. All right, Danny's going to present the results.
All right. So this is just to show that over the different folds that we had very high, nearly perfect accuracy. Actually, this is.
Yeah, so this is for step 1. So this is just the classification accuracy when we trained up the SVM for discriminating between 2-back and 0-back.
Mm-hmm. And then so these are the correlations with behavior. And so first we wanted to look at near transfer. So we have two different metrics here. This is the average n-back accuracy. So that's classed across both the 0-back and the 2-back. And we get a nice correlation here. Just looking at the 2-back trials, we still have a significant correlation between our individual dot product maps and our behavior. And then a far transfer, so both of these near transfer, these are actually in the scanner when they're doing the working memory task. And then they also completed a list sorting working memory task outside of the scanner. And so that's what this far transfer metric is here. And we see that we also see a relation. But we also wanted to assess to make sure that this could just be pure chance. So we wanted to make sure that this didn't relate to anything that it shouldn't relate to. So we just picked a random continuous variable from the HCP data set. And so we've had near transfer, far transfer, and wherever you are transfer. So they had this metric about sort of like quality of friendship. And we see here that it doesn't correlate. I mean, technically it's significant, but you can see by the value that may just be chance. Okay, so this is good. This is for our working memory signature. And as Patrick described, we also did one classifying faces versus places, and that shouldn't relate to any of our behavioral measures. And so when we did that, we have no correlations here. So that's also good. All right, so next steps. We are only presenting like a fraction of the things that we did do. We thought this was sort of like the easiest thing to talk about. So we also, one of the goals of this project was to assess like different machine learning classifiers. We did the same analysis using a continuous measure of accuracy rather than sort of dichotomous 0 versus 2 back, and have some results for that. But we'd also like to see, comparing parametric versus non-parametric or other types of classifiers like logistic regression, what could sort of generate the best predictive map or neural signature. So that's like one question that we like to pursue. Also, whoopsies, not yet, sorry. We also, so right now, we just are sort of looking at the whole map and correlating that with behavior. But one thing that we're thinking about to try to assess if like certain areas were more crucial to other regions, sort of parcellating the brain and then knocking out specific parcels, and then taking that map and correlating it with behavior to sort of try to triangulate to see like which areas are particularly important or have the strongest correlations with behavior. And then, so this was just one example, and as Patrick said, we chose working memory because it exhibits like a really, robust pattern. But you could also do this with any psychological process that you're interested in. And so that's sort of the big picture would be that this might be a useful way to relate brain activity to psychological processes. So actually using the brain to tell us something about psychology. So we'd like to pursue other processes of interest, and there's a number of them within the HCP data, but you could also do this on your own data. All right. Any questions?
Do you have any photos or like of the maps, like what the maps look like?
So we do. I mean, we didn't just because it's not looking at regression weights for classifiers isn't really can be misleading about what regions. I had a conversation with Tal about this. It's not necessarily informative about which regions truly are the most informative and can be misleading sometimes. So and the idea of lesioning came from Tal essentially to actually know which region contributes most to the model. That's a better way than just kind of visualizing the whole brain map. And that's like the kind of the theoretical region. The practical reason is that working with like Connecto and Workbench, I don't have much experience with that. It's kind of a pain in the ass, so just doing good.
Other questions?
Not bad. For the out-of-scanner thing, did you consider training directly to predict out-of-scanner from the imaging data?
So the actual behavior or the out-of-scanner memory?
The out-of-scanner, out-of-scanner behavior.
That was one of the things we did. We did a SVM regression with the 2-back task with the out-of-scanner behavior, and it worked the same way. We still got the same correlations with working memory and no correlations with the non-working memory tasks.
And actually, if you have ideas about that, because we were
comparing approaches, and for some reason this approach seemed somewhat more impressive almost, that it works when you don't train on that. But I don't know, they're different. So, yeah.
How much does in-scanner 2-back performance predict out-of-scanner age-adjusted working memory? Just behavior.
Well, you did the correlation, it's pretty high.
Oh,
But it's not super high.
no, wait, so you're saying just like behavior and behavior?
Yeah. Because that's like,
No, I did, sorry, for which measures again?
whatever, like when you're 2-back, let's say 2-back performance versus the age-adjusted.
With like memory?
Yeah.
I think it's like a 0.3 correlation.
It's a 0.3 correlation, and what if you had a
0.6 for the combined
on the
accuracy.
brain? So you can predict out of scanner behavioral performance better than the in-scanner.
Or for the memory it's like 0.5, yeah.
In-scanner better than out-of-scanner.
That's my question.
Yeah, it's about the
Sorry,
reliability
just-
of the behavioral measure.
Right,
That's
is
his question.
behavior a better predictor of behavior than the brain is?
In this case, no, but
Which is kind of interesting.
Yeah.
yeah, definitely.
Yeah, no, that's the limit to how well you can predict it.
Yeah.
If the behavior doesn't correlate with itself, then you can't possibly correlate with something else in that behavior, right?
Oh yeah, except this was a transfer. This was a different task. But yeah, it would be interesting. Like I had the same thought. Like the ceiling is how well in scanner 2-back is down or scanner 2-back.
Also, this is 2-back accuracy, not D prime, which is what most people do, so it's a little bit of a weird measure, but we didn't have D prime.
I mean, one way you can think about this is that, I mean, the classifier is basically identifying kinds of activity that discriminate recognition memory from working memory. 2-back is sort of involves working memory, and 0-back is essentially just, oh yeah, I recognize that. And so it's saying that people who show a pattern of brain activity that's more like the canonical pattern do better.
Jeanette used a very technical term. She's called more 2-backy looking brain, which means better performance.
How different are those patterns from just an average of the image of the 2-back or 0-back? And you gain a lot by doing the...
We haven't tried that yet. You mean just like a single number?
Look at, it would be like a nearest neighbor on the average. I mean, it would be like so simple and without any, how much are you gaining if you...
Yeah, definitely. I mean, we would want to sort of compare it to see if there is any like incremental value from this approach. Definitely.
Maybe as a complementary suggestion to what Tel said with deletions, since you use SVM, deletion makes a lot of sense since your weights are going to be everywhere, but try one of the sparse classifiers and try and classify once and predict, classify again and predict, and keep doing this until you actually have only random classification left. Then you get an idea of which features are informative, even if you have removed the highest weights, essentially.
Cool. Yeah, thanks.
All right.
Take a little break and stretch our legs and get off the boat. There's a company upstairs, I think.
Yeah.
Coming and do it in 10 minutes.